This conference will now be recorded.
Okay, ma. Today we are going to discuss different types of staining techniques that are of mainly differential staining techniques. Already I told in the differential staining about the gram staining technique, which is mainly used to differentiate the gram positive bacteria and gram negative bacteria. Okay, and next coming to different types of differential staining techniques are present for the differentiation of the different microorganisms based on their structural properties also. Here, based on the structural property, mainly Mycobacterium species, they can be differentiated by using the acid fast staining technique. And here, for the Mycobacterium tuberculosis identification, fluorochrome staining techniques are also used. And also for the volutin containing organisms, that means here the storage organelles are present in the organisms, and that can be identified by using the differential staining, and that mainly uses the Albert stain. Albert stain is mainly used for the differentiation of volutin containing organisms. And next, endospoor forming microorganisms. Endospoor forming microorganisms can be identified by using the malachite green stain. Okay, and next, capsule. That means here the bacteria are mainly surrounded by the outer layer called as the capsule. For the differentiation of capsule, here positive staining technique is the negative staining techniques are there. And these are mainly used for the differentiation of different types of microorganisms. And also flagella containing microorganisms and the spirochetes that can be differentiated from the spirillum, and that means these are the different types of staining techniques for the differentiation of different microorganisms, and that comes under the differential staining techniques, under which mainly acid fast staining technique is mainly used. Here, acid fast, and this is mainly used to identify the mycobacterium species. And these mycobacterium species, these are in the form of tubercle bacillus. And this acid fast bacteria, why these are calling as acid fast bacteria means, based on the structural properties, these are identified by using the staining called Gill-Nielsen staining. Here, acids are present on these structures. That means here mainly mycolic acids. Mycolic acid is present in the cell wall, and that gives the intactness to the cell wall. Okay, and here this degree of acid phosphate, and that varies in different types of bacteria. Here, this acid consists of acid fast bacilli. It is due to the presence of mycolic acid, and that is called as the unsaponifiable wax fraction, and that is present in the cell wall. And due to this mycolic acids, and these can only be stained by Gill-Nielsen stain, and that means here the normal staining processes are not used for this technique for the identification of mycobacterium. Okay, here in this staining mainly heat causes the penetration of the dye into the tubercle bacilli. Here, the main principle behind the Gill-Nielsen staining is here heat penetrating into the tubercle bacilli, and that causes the visualization of these mycobacterium species. Okay, and due to the heat fixation of the microorganisms, here once stained, the stain cannot be easily removed. Heat fixation causes the penetration of the dye into the tubercle bacilli means here, and that stain cannot be easily removed. That means if the mycolic acid structures are present in the constituents means, and that stain can be taken by that mycolic acid, and that confers the acid fastness to the bacteria. Whereas the other microorganisms and that are not uptaking these strain means they do not containing the mycolic acid in their structure. In this way, we can identify the acid fast bacteria from the non-acid fast bacteria. Okay, and here the other microorganisms which are easily decolorized by acid alcohol, these are considered as the non-acid fast bacteria. And this non-acid fast bacilli, and these they can take the other strains. That means that other strains are called as the counter strain. Here we are using two types of strains. First one is the primary strain and the second one is the counter strain or the secondary strain. In between these, and here the treatment undergoes by the acid alcohol decolorization method. In the primary strain, here the primary strain mainly used is the carbol whereas the counter strain and that contain methylene blue or any other type of strain. But here the mycolic acid it takes only the primary strain due to the mycolic acid in their structures. Okay, and in this way we can differentiate the acid fast and non-acid fast bacteria. And here these acid fast cells they retain the red color of the primary strain. Here mainly the primary strain used as the carbol and that gives the red color. Okay, and hence we can identify the acid fast cells. Okay, here these are the staining methods that can be mainly used for the identification of mycobacterium or the acid fast bacilli. Okay, mainly Gene Nielsen hot stain or Canyon's cold stain method. These are mainly used for the differentiation of acid fast bacteria from the non-acid fast bacteria. And this is the structure of acid fast bacteria, and they contain mycolic acid in their cell walls. And due to the acidic components, that is the mycolic acid components in these structures, here these are called as the acid fast bacteria. Okay, and here these are the staining methods that are mainly used for the identification of acid fast bacteria. Okay, and what are the main requirements for this means here? The primary staining, it is done by mainly strong carbol mainly used. Okay, next, decolorization with acid alcohol. Here the acid alcohol contains 3% HCl and 95% ethanol, or it may contain 20% H2SO4. Okay, and next, counter stain by using the methylene blue. Methylene blue is which type of dye? Acidic dye or basic dye? Vidushi, is it acidic dye or basic dye, ma? Methylene blue is which type of dye? I have already told about the different types of dyes. Acidic dye, basic dye and neutral dyes. Yeah, basic dye. Correct, ma? Here, this is the basic dye. Here, we are first staining with the strong carbol that is called as the primary staining agent. And next, counter stain with the methylene blue. And in between these, decolorization with acid alcohol. And here, during the observation of these cells under the microscope, the acid fast cells, these are observed as the red colored bacilli. Whereas non-acid fast ones, these are observed as the blue color. Why these are appearing in blue color means here these are uptaking the counter state. Okay, hence the non-acid fast bacteria, these are observed as the blue colored rods. Okay, and next coming to the what is the main procedure involved in this? Here, first, preparation of smear. Already you know about the preparation of smear. Just we are taking the culture and by adding a small little drop of water, or the culture is spread on the slide. That is the preparation of smear. Here, just normal heat fixing occurs for the fixing of cells firmly to the slide. Here heat is mainly used for the fixing of cells to the cell, firmly to the slide, and that is heat fixing. Here, after the preparation of spear, here the heat fix is mainly used for the identification of mycobacterium species. Heat fixation step. It is the compulsory step here in identification of mycobacterium species. Okay, then the smear is to be flooded with the primary stain, that is the carbol stain. Carbol stain. And next, after the carbol addition also, it should be steamed for about 5 minutes. For 5 minutes. And if the carbol stain is needed, then you can add more carbol stain. Okay, then after this, the slide is to be cooled for 5 minutes. Okay, then it is washed with the distilled water. Then it is to be added with the decolorizing agent, that is the acid alcohol, which containing the acid and alcohol. That causes the decolorization, and that means here it is removing the primary state after the heat fixation only, and this is to be decolorized. Okay, and next, then here acid alcohol is added in a manner such that it should be added in drop wise by tilting the slide by 45 degrees centigrade. And here, due to the addition of this acid alcohol, decolorization occurs. That means the red color of carbol it is streaming from the sphere. That means it is going away from the sphere. That means decolorization occurs on the sphere. Then it should be rinsed with the distilled water. And after that, after drying process, then the secondary stain or the counter stain is to be added. That is the methylene blue. Low plus methylene blue stain is to be added. And this stain it should be this to kept on the slide for about 15 to 20 seconds. And then the slide is rinsed and let it dry. And then next here mycobacterium species. These are visualized by using the oil immersion objective. Oil immersion objective. Okay, what is the magnification power of oil immersion objective? Magnification of oil immersion objective. What is the magnification power? Yeah, 100x. That means here it can be visualized by using the 100x, and that is the oil immersion objective. And that means if the acid fast bacteria that can stain means these are appearing as the red color. Okay, this is the total process, and that is to be done during this procedure. Here, the stained smear and that contains the pink color slender rod shaped structure. That means the dark red colored structures are seen with the curved ends. And that means here the acid fast bacteria are present and non-acid fast bacteria are also present here in this. Here, acid-fast bacteria, these are mainly observed in the form of pink-colored, slender, rod-shaped structures. Whereas the blue-colored ones, these are called as the non-acid-fast bacilli. And that means if the smear contains acid-fast bacilli means that are appearing as red color. And that is the microscopic reading about the acid-fast bacteria. Here, we already I said two methods are mainly useful for the acid fast bacteria identification. That is the first one is the this first one is the acid fast staining, that is the Gill-Nielsen staining, and second one is the Kine-Nuance method. Here in Kine-Nuance method, it is called as a cold staining technique. Why it is called as a cold staining technique means Kine-Nuance technique does not involving the heat fixation after the addition of the primary stain, and also the decolorizing agent, and that should be different from the Gill-Nielsen stain. Here in Kine-Nuance method, mainly 20% sulfuric acid is mainly used for the used as the decolorization agent. Whereas in Gill-Nielsen staining, acid with combination of alcohol is used as the decolorizing agent. Okay, and that are the differences between Kine-Nuance method and Gill-Nielsen staining. Kine-Nuance method is called as the cold stain method, whereas Gill-Nielsen stain method is called as the heat method, heat fixing method. Okay, and that are the basic differences. Okay, and also different modifications for the acid stain present for the differentiation of different microorganisms. And that means here mainly mycobacterium species are mainly used for the identification. In the mycobacterium mainly here mycobacterium tuberculosis is present and mycobacterium leprosy also there. And here 5% sulfuric acid that is used as a decolorizing agent, and that is mainly used for the staining of mycobacterium leprae that causing the leprosy. Okay, and also for the Nocardia species, Cryptosporidium species, Isospora oocyst, here 1% sulfuric acid is used as a decolorizing agent. That means the modifications are present in the decolorizing agents for the observation of different types of microorganisms. Okay, and also 0.25% sulfuric acid is also used as a decolorizing agent for staining spores. Spore staining techniques uses the 0.25% sulfuric acid as a decolorizing agent. And this is the method for the identification of acid-fast bacteria, and that is involving Gill-Nielsen staining and Kine-Nuance method. Okay, this is all about acid-fast bacteria. And also, these acid-fast bacteria, they can be observed by adding the fluorochromes. By adding the fluorochromes. That means here fluorescence microscopy. By using the fluorescence microscopy, visualization of mycobacterium species can be done. That means what are fluorochromes? That means they using the phenomenon called as a fluorescence. What is fluorescence phenomena? Do you know about the fluorescence? Fluorescence phenomena, and that mainly applicable in fluorescent microscope. Fluorescence phenomena. Actually, fluorescence means it absorbs the one wavelength radiation and emits the other wavelength radiation, and that is the main fluorescence phenomenon. Here, fluorochromes or the dyes that are mainly attached to identify the particular species here. And here, that means the fluorochrome stained smears, and these are mainly used for the observation under fluorescent microscopy, and that causes the visualization of the particular microorganisms. Okay, good ma. Here, the visualization of the particular microorganism occurs at the higher magnification levels, and that is at about 250x to 450x. That means higher magnification causes the rapid scanning of the sphere, and that causes the particular visualization of microorganisms. And here, the main fluorochromes that are used for the staining purpose are the aramine and rhodamine, and that are the fluorochromes, and that are mainly used for the identification of acid-fast bacilli. And these appear as yellow colored one against a black background. These are appearing as yellow colored ones against a black background. That means here, depending upon the microscope you are using, the technique also differences. And the colored ones are also different. The colored visualization is also different. And what is the procedure? And that is involved in the fluorochrome stain process means here also the heat fixed stain smear it is mainly used. And here the sputum that should be taken for the observation under fluorescence microscope, it should be thin sputum. And here then it should be covered with the aramine phenol stain, aramine phenol stain, and it kept at the room temperature for 10 minutes. And then the stain is washed off with the water, and then with the it is covered with the decolorizing agent, that is 1% acid alcohol is to be added, and it is kept about for 5 minutes for the decolorization process, and that causes the decolorization of the stain aramine phenomenon. Okay, and next after this secondary what we are using here counter stain is the 0.1% potassium permanganate, and then it is left for 15 seconds. Then also it should be washed, and then it should be dried. Okay, then it is observed under the fluorescence microscopy, and that containing the 4 mm objective. 4 mm objective. Here, how these are appearing means these are appearing as the yellow colored rods and in a dark field, in a dark field. That means here also oil immersion objective is used for the detection. Okay, and here these are observing as the yellow luminous rods under a dark field. And then there is the basic difference between the particular microscope used for the visualization of the images. Okay, here different staining processes are present for the for the differentiation of different microorganisms. Here fluorochrome labeled stains are used, and the normal staining procedures are used for the acid fast bacteria differentiation. And this is a differentiation of acid fast bacilli with non-acid fast bacilli. Here these are also observing as the rods, but these are in yellow colored ones. These are yellow colored rods against a dark background. Okay, and this is called as a fluorochrome stain. And next differential staining technique is the volutin containing organism identification by Albert stain mechanism. Albert stain mechanism. Here, that means the volutin means what are the volutin granules? These are the storage granules. Here, volutin means these are the polyphosphate granules or the metaphosphate granules, and that are present in the microbacterial cytoplasm. These can be stained by using the different staining techniques. Okay, how these are observed when these are stained means these can be seen in unstained wet preparation as the round refractile bodies. This can be seen as unstained wet preparation as round refractile bodies when these are observed under the microscope. But some Albert strain is mainly used for the visualization of these granules. These granules. And here, for the staining of these volutin containing microorganisms, mainly basic dyes are used. Basic dyes are mainly used. What are the examples of basic dyes? Examples of basic dyes. Yeah, methylene blue, saffronin, crystal violet. These all are called as the basic dyes. And that means that are the positively charged radicals. And that connects with the negatively charged particles present in the cytoplasm. Okay, and that means here basic, yeah, methylene blue, toluidine blue, thionin, crystal violet. Okay, ma, good. Here, basic dyes. These are staining more strongly the volutin containing organism than the rest of the organisms. Okay, and that causes the hematocyte. Okay, and these are observed as the reddish purple color, reddish purple color. And special methods are present for the identification of these microorganisms that are of mainly Albert stain and Neisseria stains. And these causes them dark purple color. And but the remaining bacteria, these are observed as the contrasting counter stain. Okay. Next here, here Albert stain, Neisseria stain. These are mainly used for the identification of volutin containing organism. Mainly, it causes the identification of diphtheria bacillus. Diphtheria bacillus. Okay, and that are mainly identified by using them culturing on blood or serum media, or different types of media are used for the visualization of these microorganisms. Okay, what is the procedure that is involved in these ones? Here, first, normal process, that is the making of the smear, and that is the smear preparation next. After the preparation of smear, and these to be dried in air, and it is heat fixed. Then it is covered with the all but stain, and allowed to act for 3 to 5 minutes. That means the stain is to be placed for about 3 to 5 minutes, and then it is to be washed in water, and then it is to be dried. Then it is to be added with the Albert's iodine, Albert's iodine, and allow it to act for 1 minute. Okay, here what is happening? Albert's iodine, and that is the dark blue color, that is bluish black color. And that means here, when it is stained with the Albert's iodine, means that giving the granules that are staining with that color, and these are appearing as the bluish black color, and the protoplasm appearing as the green color, protoplasm appearing as the green color. And if these cytoplasm containing granules are present, then only this bluish black color appears. When the other organisms present, that means they lack that metachromatic granules or the polychromatic granules, means these are appearing as the light green in color. And by this way, we can identify the volutin containing microorganisms from the non-volutin containing organisms. Okay, and that is the method involved in mainly in this. Here, Albert stain is mainly used for the visualization of volutin containing microorganisms. And here, this Albert stain, what is this contains? Albert stain. Albert stain mainly contains the basic dyes that are of mainly toluidine blue and malachite green. Malachite green. And that causes the high affinity with the acidic tissue components present in the cytoplasm. And that means here these are the basic dyes. These basic dyes are mainly used to identify the acidic components. Acidic components means that are the volutin granules. Okay, in this way, these are observed. Okay, and that is the main procedure and mechanism involved in Albert stain. Here, Albert stain mainly used for the identification of diphtheria, Corynebacterium diphtheria, and these are appearing as the colored bacilli, and that showing the Chinese letter arrangement. Chinese letter arrangement at angles to each other containing bluish black metachromatic granules of cell. These are the metachromatic granules and appearing as the Chinese letter arrangement. Chinese letter arrangement. This is the important point mark for any test. Here Albert stain, and that mainly causes the identification of volutin containing microorganisms that are of mainly Corynebacterium diphthi, and these are appearing in the form of Chinese letter arrangement, and that are observed in the form of bluish black metachromatic granules. Okay, and this is called as the Albert stain mechanism. Okay, next coming to the spore stain. Spore stain. Here, spores that are the endospores, these are the resistant. That means during the unfavorable condition, some of the bacteria they forms the spores. Okay, and this morphology of the bacterial endospores, these are observed in unstained wet films, and these are mainly observed under the phase contrast microscope. And what is the basic principle involved in phase contrast microscope? Phase contrast microscope. Here, phase contrast fluorescence. Okay, and that is the phase contrast microscope. Here, the spore staining, that means the spore containing microorganisms are mainly observed by using the phase contrast microscopy. Okay, I have already told different modifications are present in the decolorizing agents. Here in the spores, mainly 0.25% sulfuric acid mainly used as the decolorizers, and that is the red spores in blue stained bacteria. Okay, here also sometimes the lipid granules, the granules that are present in the cytoplasm, these are also staining the red color, and these are also appearing like small spherical spores. And that means here we have to differentiate between these also. That means the spores and lipid granules, these are appearing in the same structures. Hence, the resolution is to be based. The resolution, that means here we are using the phase contrast microscope. Okay, and what is the main method that is involved for the spore staining is here Fulton malachite green staining technique, and that is mainly used for the staining of spores. Here, method of Scaffer and Fulton, and that is called as a malachite green stain for spores. Malachite green stain is mainly used for the spore identification. And here, malachite green is a acidic dye or a basic dye. It is a acidic dye or a basic dye. Malachite green is a which type of dye? Yeah, acidic dye. Here, acidic dye is mainly used for the identification of basic components. That means here this is the negatively charged acid radicals are present, and that are mainly used for the positive ones, for the identification of positive ones. Here the spores are mainly identified by using the acidic dye. Here what is the process that is involved? Just the normal process, that is a staining process that is involved here. Okay, then the films are dried and fixed with the normal flaming, that is the film fixation. And next here, what is the difference between this process is here? The slide first it is placed on the beaker of boiling water. Boiling water. That means the upper side of the slide that contains the bacteria film. Whereas at the lower of the slide here, the boiling water that causes the vapors. And here, after that, within several seconds, what is happening? The large droplets that are caused due to the boiling, these are present, these are condensed on the underside of the slide. Then it is flooded with the 5% aqueous solution of the acidic dye that is called as a malachite green, and then it is left for 1 minute while the water continues to boil. And then after some time, that means after 1 minute, then it is washed in cold water, and then it is to be added with the 0.5% saffronin or 0.05% basic vaccine for about 30 seconds. And what is happening here means, here what is happening here, the malachite green is mainly used for the staining, means here that appearing as the green color. That means here the stain is received by the spores due to the boiling nature. And here that causes the spores to be absorbed in the form of green color. Whereas the vegetative bacilli, these are appearing in the form of red color. Whereas the granules that are present in that causes the disturbances. That means here these are not staining with that, and that means these are appearing as the unstained ones. And in this way, we can identify the spores. The spores are identified in the form of green color. And that method, malachite green stain, is mainly used for the spore containing microorganism. Here, these are appeared as the green color, whereas the saffronin stained ones that are appearing as the red color. Green colored ones are the spore containing microorganisms. Okay, and that is the spore staining technique that is mainly uses the Fulton malachite green staining. Fulton malachite green staining. And next method is the capsule staining. What is capsule? It is surrounding the bacterial mite. It is a protective layer, and that causes the prevention of penetration of different chemicals into the cell. Here, that capsule, it also to be stained. That means by for the identification of capsule containing microorganisms, the capsule staining is mainly used. And it is also a type of differential staining because here it is not using, that means the materials that to be used for the capsule means that is to be different, because here what is happening means the capsule staining is a difficult technique because the capsular materials present in the present that are surrounding the cell, and that are mainly water soluble. That means it can be removed by normal washing techniques. Hence, here the heating techniques that are mainly used for the fixation, these are not to be used for the capsule staining because it causes the disruption of the capsular material. And that means here due to the cell shrinkage that causes the clear zone around the microorganism. Here, due to the fixation by heat, here what is happening? The capsular material surrounding the cell that causes the shell shrinkage that may create a clear zone around the organism. Hence, during the capsule staining, it should not be heat fixed. It should not be heat fixed. And also the dyes that are used for this process are also different from other staining techniques, because here the capsule is the non-ionic one. Capsule is the non-ionic one. Hence, the common dyes that are used will not bind to it. Whereas the two dyes, the combination of the acidic and basic dyes, these are mainly used to stain the background and cell wall. Here, acidic dyes and basic dyes both are used for the staining the background and cell wall respectively. Okay, here the background means that can be stained by using the acidic ones, whereas the cell wall is stained by using the basic dye. And that means here for the capsular staining, the sphere should not be heat fixed. And also only one of the acidic or basic dyes. These are not to be used for the staining of the capsule. Okay, next, two staining methods that are mainly negative staining and positive staining techniques. These are mainly used for the identification of capsulated microorganisms. Identification of capsule containing microorganisms means mainly positive staining technique and negative staining techniques are used. Okay, and here what is happening means positive and negative stainings. These are mainly used for the staining of capsule. Here capsulated bacteria, and that mainly contains the polysaccharide capsule, polypeptide capsule. These are present in the capsulated structures, and basing on that we can differentiate the different types of microorganisms, and also make FDN reaction are also used for the capsulation identification in the identification of these capsulated bacteria. Okay, mainly negative staining technique and positive staining techniques are mainly used for the identification of capsulated bacteria. Okay, here first coming to the positive stain. What is positive stain? Here first normal preparation of the smear, and that means here mainly observing the capsulated bacteria that are the Streptococcus pneumoniae are mainly used, and here just these are placed on making the smear and allow it to dry, and next and then it is flooded with crystal violet, and here depending upon the process also here timing for the staining process is also different. Here, for uptake of crystal violet, it should take 5 to 7 minutes. And then water smear with the 20% copper sulfate solution. And then it is to be dried. And next, it is observed under the low power objective and oil immersion objective. Okay, and here in the culture smear, the capsule is seen as light blue in contrast to the deep purple color of the cell. During the positive staining, the capsule is appearing as the light blue, whereas the cell is appearing as a deep color, deep purple color. What happening here? The positive stain mainly it colors the body of the cell. Only it colors the body of the cells, but not the capsule. Okay, here what happens here? It colors the body of the cell. That means the body of the cell is appearing as the deep purple color cell. Whereas the capsule, it is observed in light color. It is appearing as light color. Okay, and that is the main phenomenon that is observed under the positive staining. Positive staining. Whereas coming to the negative staining, in negative staining, what is happening means here also first the slide is to be placed with the Indian ink on the slide. Indian ink on the slide. Here, what is happening means here, then the culture is to be added to the Indian ink. Indian ink means just generally it is in dark blue color. It is in dark blue color. Then it is to be placed with the cover slip. That means mount method. Mount method is used for the here for the making of the preparation of the sphere. Here, then it is placed with the cover slip. Then the ink drop is placed on that. Then the film becomes very thin. That means by placing the cover slip, it causes a disaggregation of the cell. That causes a very thin film, and that appearing as a pale in color. And it is observed under the 40x object. 40x object. And what happened here means due to the negative staining method in capsular staining, here the capsule is observed as the clear refractile body around the organism. That means just clear hollow ones appearing against a black background. What happens here is during the negative staining process, it colors the background but not the capsule, but it is leaving halo around the each cell. See the difference between positive and negative. In positive staining, what is happening? It is coloring both the body and capsule. But here, the body is appearing as in darkest color, whereas the capsule is appearing as a light color. Whereas in negative stain, what is happening? In negative stain, it colors the background, but not the capsule. But it is leaving a halo clear refractile around the organism against a black background. That is here the coloring materials are not used for this. Chromophores means the coloring ones. Here these are not used for that. But here these are appearing depending upon the staining process. That is a positive staining process and negative staining processes are used. Here just only the Indian ink is used for the observation, but it appearing in the color differences. How these are appearing is depending upon the staining process. It does not depending upon the color also here. What is happening in negative? Only just a clear refractile halo around the organism is appearing. Whereas in positive staining, that means the light and dark color is depending upon the stain. Okay, and that is the positive and negative. This is the here capsulated bacteria in Indian ink. These are observed in the form of this. Here, this is called as the positive. Whereas in negative staining, here the halo cleared ones, halo refractile body appears against a black background. Okay, and that is the difference between positive stain and negative stain. Okay, and this is the capsule staining technique. Capsule staining technique mainly includes the positive and negative staining techniques. Okay, and these can be observed through electron microscopy also. Transmission electron microscopy is also used for the observation of the capsule containing microorganisms. Okay, and then we can go for staining of spirochetes. What are spirochetes? Here, the spirochetes, the three important groups of spirochetes are present. Here, these are of mainly Trypanoma, Leptospira, and Borrelia. These are the main spirochetes. Here, these are the motile, elongated, flexible, and spiral organisms. These are the motile, elongated, flexible cell wall is present in spirochetes. Flexible cell wall. Flexible here means flexible cell wall. These are in spiral shape and containing flexible cell wall, and motility is present. Motility here mainly by axial filaments. Motility may be due to the polar flagella and by using the axial filaments. Here, motility is in spirochetes is mainly by axial filaments. Axial filaments. Difference is present in between the spirochetes and spirillum. Here, in the spirochetes are the spiral shaped organisms, but the flexible cell wall is present, and the motility mechanism is also different. Motility is by mainly axial filament, whereas the spirilla or spiral shape, but they contain rigid cell wall. Rigid cell wall, and the motility is by mainly by using the polar flagella. Polar flagella. Here, these are not easily stained, and also here the spirochetes, these are not easily observed by the bright field microscopy. Here, these are not directly observed by the bright field microscopy. Here, these are not easily stained ones by normal ordinary methods. Ordinary methods. And here, the Trypanoma species and Leptospira species, these are very two things to be demonstrated by ordinary stains. To be demonstrated by ordinary stains. Whereas these are observed only under the dark ground microscope. Dark ground microscope, that is the dark field microscope. Differentiation is present in between bright field and dark field. In dark field, what is happening? Dark background causes the brightest image. Brightest image is observed under the darkest background, and that is the dark field microscopy. Here, the spirochetes, these are observed by using the dark ground microscopy, where here the bright appearance causes the visualization. And here, for the visualization of these spirochetes, permanent preparation is to be done. And it is mainly by using the silver impregnation method. Silver impregnation method. In silver impregnation method, mainly by using the deposit of silver, here impregnation is to be done. That is, a permanent preparation is to be done. The main methods for the fixation, the permanent preparation are the Fontana method and Liavaditi method. Liavaditi method. Mainly Fontana method is used for the visualization of spirochetes. Here what is happening means, first it should be the film, that is the sphere, that should be made with the, that means the fixative is made, and it is 30 seconds each time. Here treat the film 3 times 30 seconds each time with the fixative. Here the fixative is the silver one, and that means here we have to make the first film, and then here the fixative is to be washed with the absolute alcohol and allow the alcohol for 3 minutes, and then here excess alcohol that is added to be removed, and the film is to be dried. And next here here, then pour on the mordant and heating till steam rises and allow it to act for 30 seconds. And that means here the fixative that is added in high amounts, and that is to be added in high amounts. And next it is to be washed, and then it is treated with the ammoniated silver nitrate, and then it is to be heated till steam rises for 30 seconds. That means here the total film, that means the sphere that is to be observed, and that is to be in the form of brown in color. Brown in color. Then it is to be washed with the distilled water, and then mount with the Canada balsam stain. Canada balsam stain. And next, then it is to be placed with the cover slip. Then it is to be observed under the oil immersion ones. Here, the stain, it is taking the more amount of the ammoniated silver nitrate. That means the silver nitrate that is appearing as brownish black color. Brownish black color. Here, the spirochetes are stained in the form of brownish black color on a brownish yellow background. It is the here brightest image is observed on a dark background, and that can be observed. That is, a dark field microscopy can be used for the visualization of these microorganisms. That are the spirochetes. Okay, and that is the here the leptospies from a culture stained by Fontana silver technique, and that are examined by dark field microscopy. And here these are the brightest colored ones, that are the dark browny and black colored ones. These are appearing on the dark background. Okay, and that is called as the Fontana's microscopy, and that are mainly used for the identification of spirochetes. Okay, next coming to the here spirochetes only containing the axial filaments for the motility. whereas the flagella containing microorganisms are present and that also can be visualized, and these are mainly visualized by using the electron microscopy. Electron microscopy. Here, the flagella, these are visualized by electron microscopy due to the thickness that is increased by adding the fixative. Okay, and different bacteria contains different types of flagella. That means depending upon arrangement of flagella, the bacteria are classified into peritrichous, locotrichous, amphitrichous, atrichous flagella. Here, that means the flagella, these are to be identified, and that means different staining techniques that can be identified for the, that can be used for the staining of flagella, and it is the main locomotory organ that causes the locomotion. Here, for the flagella staining, mainly wet mount technique is used. Wet mount technique is used. And it is mainly first described by Heimbrook. Heimbrook. It is the wet mount flagella stain that was mainly described by Heimbrook. Okay, and here what is the procedure here involved means, here we have to observe the locomotory organs means, and that means here the locomotion is to be present. That means these are to be active during the staining process. These are to be active. That means the motile bacteria are to be identified. That means the bacteria is to be grown on a non-inhibitory medium. That is, the growth is to be not to be stopped. That is, the motility is not to be stopped during the observation. And that means here the bacteria should be grown for 16 to 24 hours on non-inhibitory medium, and that are of mainly tryptic soy agar or blood agar. Tryptic soy agar or blood agar. And next, water is to be placed on the colony. That means here we are transferring the culture, and on the culture just a loop full of water is to be added, and that causes the motile bacteria to swim. Motile bacteria to swim. And then here this loop full, then it is to be transferred into a loop full of water on a side to get a turbid suspension, and then it is to be covered with the covers, and that causes the just a small agitation, that is the movement. Agitation means the mechanical power due to the placing of the cover slip, that causes the movement, and that causes the detaching of the flagella. That means here the flagella that is present on the structures, and that is detached by using the agitation, that is the mechanical force to be applied, and that means here cover slip is placing on the suspension, and that causes the detachment of flagella. And here after 5 to 10 minutes, then reuse stain is added to the cover slip. Reuse stain, and it is used for the to diffuse into the film, and that causes the observation of flagella under the microscope. Mainly used stain is mainly used for the identification of flagella containing microorganisms. Here, the Salmonella typhimurium, it contains the mainly flagellated ones. It is a type of peritrichous flagella. And here the peritrichous flagella that are arranged around the cells, and that are absorbed by using the reuse stain. Okay, and this is the flagella staining technique, and that is mainly useful for the identification of different flagella containing microorganisms. And that means here we are learning about the differential staining techniques for the identification of different microorganisms. That means acid fast bacteria and non-acid fast bacteria. Next is polyethylene containing microorganisms, and next spore containing microorganisms, spirochetes, and flagellar containing microorganisms. That means the differentiation is present due to the staining process, and that causes the visualization of particular microorganism. And further, different types of stains are present. Okay, and this is the concept under the differential staining process. Okay, and I think all of you understood this process. Okay, any doubts regarding these ones? Is it clear? Vichi, Darshana, is it clear, ma? Okay. Darshana? Okay, ma. Okay. Then we will go for the questions. Okay. Here, during the staining, the smear is heat fixed in order to. What are the options? What is the main option? Actually, in acid fast staining, heat fixing is the main principle here. Just in normal process also, some amount of heat fix is to be done. And why we are using the heat fixing for the smear preparation? Why? To kill the organisms or to attach the organisms and kill the organisms and attach the organism firmly to the slide. Which option is the correct one, ma? Yeah. Good, ma. Vidishi. See? No, no. Darshana, no, ma. Killing of microorganism is not the correct one, ma. Okay. Here what is happening? This is heat fixed. Heat fixing means that causes the attachment of the organisms firmly to the slide. Heat fixation causes the attachment of the organisms firmly to the slide. And during this process, it is not killing the organisms. It is not killing the organism, but it is not killing the growth parameters only. That means here B option is the correct one, ma. B option is the correct one. Okay, and next, second one, the colorization of bacteria with more than one dye solution to distinguish between microbial cell types. It is called as more than two dyes are used for the distinguish between microbial cell types. It is called as Priyama Kumari C. Yeah, good ma. Differential stain. Differential staining techniques are mainly used for the differentiation of different microorganisms. Here, more than one dye solution are used. Primary stain is used and counter stain or the secondary stain are also used. Okay, next coming to the third one. What do negative staining and positive staining have in common? They stain the specimen, they stain the background. These are used only for light microscopy and these are increasing the contrast between specimen and the background. Which option is the correct one, ma? Negative staining and positive staining. Yeah, not ma. What is negative staining and positive staining? What is happening during these stains here? Mainly negative and positive staining are mainly useful for the capsular identification. And in the positive stain, what happens? It colors the body of the cell. It colors the body of the cell, not the capsular one. But the capsular appears in light color. That means here it is not staining the specimen, negative stain, and that is clear now. And next, also background. Background, that means the positive stain that colors the body of the cell, but not the background. Whereas negative stain colors the background. Negative stain colors the background. See the differences in between negative and positive staining. What happened in positive staining? Positive staining colors the body of the cell, not the background. And also it is not staining the capsular ones. And in negative stain also, in negative stain, it colors the background, but it is not staining the specimen. It is not staining the specimen. That means second option is also not correct. Okay, and these are used only for light microscopy? No. Here the capsulated microorganisms, these are not used for light microscopy. Okay, they increase the contrast between specimen and background? No. Here we are not observing the contrast between specimen and background also. Here they have colorless chromophores. That means here the colored chromophores means the colored substituents are not we are using here. Just we are using the normal staining procedure, that is the Indian. But here that means here we are not using the color, we are not using the colored one. They have colorless chromophore. That is the common in between positive and negative stain. That means for the third one, E option is the correct one. See the differences here. Negative staining and positive staining. Here they have only colorless chromophores are used for the staining of capsular material. Okay, is it clear, ma'am? Third one. Is it clear? Yeah, okay. Next, which of the following stain is used for visualizing mycobacterium? Mycobacterium. Yeah, D is correct one. Here, negative stain is used for the capsulated ones, endospore mainly for the spore containing ones, gram staining for the differentiation of gram positive and gram negative. Okay, okay, is it okay? Next, spirillum. It is not classified as a spirochete because spirochetes. I have already told the differences between spirillum and spirochetes. What is the main difference? Spirochetes possess which ones? Yeah, B. Correct, ma, Vidushi. Spirochetes possess axial filaments. Spirochetes possess axial filaments. Whereas spirillum possess flagella. Yeah, spirilla have polar flagella. Good, ma. And next, sixth one. Which of the following is not a characteristic of spirochetes? Spirochetes. These are also gram negative ones. Okay. Which option is the correct one, ma? For the sixth one, which of the following is not a characteristic of spirochetes? Here, spirochetes are to be, these are not directly observed by the bright field microscopy. This is the A option is the correct one. For this, here, these are the gram negative ones and these are the helical shape. We observed with the bright field microscope. It is not a characteristic of spirochetes. A option is the correct one now. They can be easily observed with the bright field microscopy. Okay, this is also the characteristic and these three are the correct ones. Okay, and next seventh one. If you know you have a pure culture of spore forming bacilli, that means here bacilli are forming the spores, but the bacilli here the endospore stain on sample shows only pink rods. Pink rods means here the endospore staining causes the pink rods. That means bacilli are observed in the form of pink color. And here which is the reasonable explanation for what you see? What is the explanation for this? Here, pure culture or spore forming bacilli is present. But here, the endospore stain on sample shows only the pink rods. Only the pink rods. That means here, what is the explanation for this? Here, the cells are transmitting the pink light or any spores are transmitting the light. Which option should be the correct? Simple one, ma'am. Think once. Actually, here what is in the question here? Pure culture of spore forming bacteria is there, but it is producing only pink rods during the culturing process means, and that means at the time of producing that staining process, here the spores are not produced at the time. Spores are not produced at the time. That means the spores are produced during the unfavorable conditions only. That means here during the staining process, the endospore staining process, here only the pink rods are observing, not spores are observing. That means here the spores are not formed at the time during the staining process. Okay, that means C option is the correct one. Just logical thinking. Okay, and these are the type of questions and that to be included in staining techniques. Any doubts, ma'am? Any doubts? Is it clear, all of you? Okay, okay, ma'am.